Hi, my name's Nick, and I'll be your host for this week's Galaxy Show. Today we're going to be looking at extremophile bacteria, but before we get into any details, we need to define what a bacteria is. A bacteria is a single-celled microbe. They contain no nucleus and no membrane-bound organelles. These organisms tend to thrive in pH levels of around 7 and an optimum temperature range of about 37 degrees. Now, when we're looking at extremophiles are organisms that thrive under extreme environmental conditions. Such conditions will be covered later on in this program, and they will cover a range of pH levels and temperature levels. And now I'll pass you on to Lexi, who will be interviewing Dr. Matt covering pH extremophiles.
Thank you, Nicholas. Lexi here at Yellowstone National Park with special guest Dr. Matt from the Extremophilic Institute of Australia. Welcome to Galaxy.
Hey Lexi, thanks for having me.
So Matt, what kind of extremophiles live in extreme pH conditions?
Well Lexi, there's two main branches of extremophiles. There's acidophiles and alkalophiles. Acidophiles, which are able to survive in low pH conditions of around 2 pH and even lower. They're often found in acidic locations like volcanoes, sulfur pools, geysers, and even in our stomachs. Alkalophiles live and thrive in environments at the opposite end of the scale and are highly alkaline conditions of a pH of 9 or higher, often found in places like hot springs in the Yellowstone Park, soda lakes, and the bottom of the ocean.
So how do they survive in these hostile environments?
So Lexi, generally speaking, both acidophiles and alkalophiles have cellular machinery which either requires or functions optimally at a neutral pH of around 7. So they need a method of either raising or lowering the internal pH of the cell.
So how do they do that?
They do this through two ways. There's a passive method and an active method. The specifics between the two vary. Passive methods of regulating the internal pH of both acidophiles and alkalophiles involve making the use of large molecules embedded within the cell wall, which create to form a matrix or a shield which either slows or blocks some external molecules from passing into the cell.
That's really cool.
Some acidophiles have been noted to secrete a thin biofilm which slows the passage of molecules from within the cell. The passive method, however, is not enough in either organism to maintain an optimal internal pH, and this is where the active methods come in.
So what is the active method?
The active methods of regulating the internal pH require a pump embedded in the membrane in both acidophiles and alkalophiles. They use these pumps to keep the pH neutral though it's used in different ways. Acidophiles use an active pump to remove hydrogen ions from the cell cytosol and release them outside of the cell, which causes a raising of the pH levels within the cell. The pump in alkalophiles, however, takes the sodium from within the cell in order to pump hydrogen ions from the external environment into the cell. More hydrogen ions causes the cytosol to become more acidic and therefore more neutral. Though acidophiles and alkalophiles thrive in completely different environments, they utilize very similar techniques in order to maintain their internal cell pH at a level in which they operate optimally.
Okay, thank you so much for all of that. That was amazing.
No problem, it's my pleasure.
Okay, thank you. Okay, now back to you, Nick.
That was very interesting stuff, Lexi. Now we're going to go to another part of Yellowstone National Park where Ali will be interviewing Dr. Brody, and he will be covering extremophiles concerning temperature.
Thank you, Nick. Ali here at Yellowstone National Park with special guest Dr. Brody from Extremophiles Institute of Australia. Welcome to Galaxy.
Hi Ali, thanks for having me.
Okay, so what exactly is a thermophile?
In order for me to answer that question, I'm going to refer to what's over there. Here we can see one of Yellowstone's most fascinating geothermal springs, a water body that reaches surface temperatures in excess of 70 degrees Celsius and appears almost lifeless. However, prokaryotes, and in particular bacteria, are actually present in this extremely temperate water body. These thriving bacterium that can grow and reproduce in such high temperature environments, such as 40 degree to 90 degree ranges, are coined thermophiles, i.e. temperature loving. One of the most well-studied thermophilic bacterium is Thermus aquaticus, in which inhabits these very geothermal springs and lives quite happily at 70 degrees Celsius.
Okay, so what makes these thermophiles so special? How could they possibly survive at such extraordinary temperatures?
A thermophile's survivability at such temperatures ultimately comes down to the strength of their bonds found within proteins and enzymes. Thermophilic bacterium proteins have evolved to occupy greater disulfide bonding and ion pairs that require a larger amount of energy input, in this case, thermal energy, needed to pull them apart, and hence denature the protein. Also, the increased presence of salts helps to protect the phosphodiester bonds in the DNA backbone, allowing for DNA replication at higher temperature ranges. The highly heat-stable DNA polymerase, isolated from Thermus aquaticus, called Taq polymerase, has heavily impacted the biological community, particularly as it has enabled the use of PCR, polymerase chain reaction, a very useful DNA amplification method.
Wow, that's interesting. If there's bacteria that can survive at high temperatures, could there also be bacteria out there that enjoy very cold temperatures?
There definitely is. These types of extremophiles are called psychrophiles and typically thrive and reproduce within a temperature range of -20 to 10 degrees Celsius. An example of a psychrophile, called Palaromonas vaculata, lives underneath the sea ice found in Antarctica and has an optimal temperature range of 1.5 to 4 degrees Celsius. A very important factor of how this particular psychrophilic bacterium can survive at such low temperatures is reflected in the concentration of fatty acids found within its cell. These fatty acids act as an antifreeze mechanism, supporting life at an unusually low temperature.
Thank you for your time and very informative insight to both thermophiles and psychrophiles, Dr. Brody. Back to you, Nick.
Thanks, Sally. That was very informative. We got to cover extremophiles living in different environments such as pH and temperature. Well, unfortunately, that's all the time we have for this week, but please stay tuned to our next episode of Galaxy. Till then, goodbye.